So hello everybody and welcome back to my YouTube channel, and in today's video we will be discussing about the different subjects and the course curriculum syllabus that is being taught in NIPER. So I'm currently pursuing my MS in pharmacology and toxicology from NIPER Mohali. So total there are 9 subjects, 1 credit presentation, 4 labs in the first semester for the pharmaceutics field and pharmacology field majorly. Rest also might be having similar 7 or 8 subjects equivalent. So there are 4 labs for us: computer application lab, whereby we are being taught about docking and the importance, like it is linking PI somewhat, pharmacoinformatics, about molecular screening, molecular docking. Then we have biotechnology lab, whereby we learn about electrophoresis, SDS page, agar rose electrophoresis, blotting, western blotting and all. And then we had 2 core labs. So in these laboratories we learn about different other technologies and techniques that we use, like tissue processing, histological studies, bioassays, ED50 values, PA2 value, PD2 values, all of them, protein estimation, blood glucose estimation. So we learn about different core subjects in the core labs. Now if I talk about the goals of the curriculum design, how is the curriculum designed? So it is designed in my mind of keeping a research and an industry oriented both. So it's an amalgamation of research and industry orientation, right? So it's basically about to focus on upon the emerging areas, and the syllabus is continuously revised. So there are certain areas, certain cognosy drugs, and there are certain techniques, technologies whereby in order to stay with it and cope up with it, monoclonal antibodies, controlled drug delivery systems, new cellular diagnostic and imaging techniques, so these are being taught and continuously added into the syllabus, and to provide scientific footing of traditional medicines and bring about scientific and sociological aspects about pharmacy. So subjects are basically divided into 2 parts: core and non-core. So in core we had chemotherapy, pathophysiology, general pharmacology, experimental pharmacology, and in non-core we had biostatistics, separation techniques, which is equivalent to analysis plus pharmacognosy, biotechnology, biopharmaceutics, and intellectual property rights (IPR). So this is the image of that is including all the different courses: pathophysiology, general pharmacology, experimental, chemotherapy, separation, biopharm, biotech, biostats and all, and general lab experience and seminar. And if I talk about semester 2, so in the semester 2 we have drug metabolism, pharmacological assays and screening, CNS and respiratory pharmacology, autonomic cardiovascular, blood, renal, gastrointestinal pharmacology, autocoids, endocrine system and immunopharmacology, clinical pharmacology and regulatory toxicology. And then we have good laboratory practices, GLP seminar, and that is the same thing, and that is project presentation basically, and then general lab experience in the area of specialization. So there are total 16 credits in semester 1 and 14 credits in semester 2, which accounts to 30 credits of the first year, right? And in the third and final year we do not have as such any syllabus. We are being research oriented, so we have to spend our times in laboratories. So in IPR, what was taught to us? In IPR, basically the history of IPR. How did IPR evolve? What is the transformation of IPR? That what is the need of IPR? Why is the patent protection needed? Why is the GI, trademarks, copyrights needed? Then a difference between patent, copyright, trademarks, trade secrets, geographical indications, PPVFR, that is plant protection, variety and farmers right and all. Then how do you file a patent? What are the uh, different things that are included in the patent and what cannot be patented, all those information. How to file a patent in foreign? So for that we have PCT, patent cooperation treaty. So PCTNP, that is national phase. In this particular we can file in foreign, and we can get FFL, foreign filing license, and so on and so forth. So intellectual property rights was based upon these particular topics. Biostatistics, it was based upon, of course, central tendency, mean, median, mode, dispersion measures like standard deviation, range, variance, coefficient of variance and all. So measures of dispersion. Then parametric and non-parametric test. So in parametric test, we had t-test, ANOVA test, z-test. And in non-parametric test, we had a Wilcoxon, a Wilcoxon, Mann-Whitney, Friedman, Kruskal-Wallis, and other tests that are in non-parametric fields, right? Besides this, we also had studied about the population and sampling. How do you take a sample? What are the different sampling techniques? How do you avoid biasness in this particular sample? So there are probability sampling, non-probability. Then we also learned about probability and sampling distribution. So in sampling distribution, basically there is normal distribution, Poisson distribution, binomial distribution, different distribution curves that are being available. So sampling distribution is very essential. Now, why do we need biostatistics? So when I talk as a point of view of pharmacology, so suppose we get a particular data, we have two different groups like control and suppose standard or test and control. So when we compare them, we need a statistical validation. That is, I need t-test if it's dependent, if it's independent, and z-test or something like that, or unpaired t-test. And if there are more than two groups, like there is sham operated, per se operated, so we need for that as well different tests like ANOVA. So that is what is essential for us, that we obtain a comparison. We obtain a p-value, the critical value, so that we can get an idea about what is the actually statistical data is, and we can get validation for the same. Separation techniques. So if I talk about separation techniques, in this we are taught the concepts of chromatography, extraction, spectroscopy, right? It's like MALDI TOF, that is matrix associated laser desorption ionization, time of light. So spectroscopy, that is part of mass spectroscopy. Then in chromatography, we are talking about various chromatography like HPLC, GC, adsorption, partition, paper. Then there is hyphenated techniques like HPLC, MS, HPLC, MS, NMR, HPLC, MS. So there are different techniques that are short column chromatography, flash chromatography, vacuum chromatography, counter current chromatography. So these are different chromatographic techniques. And then there is separation of biomolecules. So biomolecules are basically separated by three techniques: ion exchange, size exclusion, and affinity chromatography. So why is this being taught? And in extraction, we learned about conventional as well as the novel methods. So maceration, percolation, digestion, decoction, infusions. These are all conventional. Novel methods include supercritical fluid extraction, microwave assisted extraction, and all. So why are we being taught about this? So when we have to like validate the results as well as obtain interpretation, so HPLC chromatogram is very essential. So we as pharmacologists also need a part of need a knowledge about the HPLC. How do we read the graph, a chromatogram, and all? So that is what is essential when we speak on biomolecules. So separating proteins, separating different antigen antibodies based upon their affinity with a particular ligand, an ion exchange based upon the separation based on the charges. So that is what is important, and why this is why the subject is being taught. Biopharmaceutics, of course, it's like a semi-core for us. So biopharmaceutics, may we learn about PKPD, ADME, what is the clearance, what is disposition, what is volume of distribution, half-life, different phases of metabolism and modes of absorption. What are the different parameters that affect the distribution, affect the absorption, and how can you enhance the absorption, how can you increase the bioavailability. So biopharmaceutics is all about your pharmacology, pharmacokinetics, pharmacodynamics, and all. So it was all being taught to us, that sir explained in detail about everything. Biotechnology. So in this subject, we were talking about the role of genomics in drug discovery. So nowadays we have proteomics, we have metabolomics, we have transcriptomics. So these are different types of omic techniques that are being used. So that is why a glimpse of this was given. PCR, RT-PCR, vaccines and immunology. Then we were talking about edible vaccines. fermentation, recombinant technology, so different vectors, different ligase enzymes, these were being like taught to us about various vectors available: plasmid, cosmid, bacteriophage, and all different enzymes like DNA ligase, DNA polymerase, RNA primase, and all. Fermentation may process all the bioprocess steps like upstreaming, fermentation, downstreaming, different process of cell disruption and downstreaming, recovery process, that once you get the product, because nowadays vaccines, antibiotics, all of these are obtained, they are biopharmaceuticals, right? They are all biotechnological products. So that is why this term is now evolving a lot. Antibody fragments, you have different ADC, antibody drug conjugates, that are now being available. So these are very important topics that were being taught in biotechnology. Pathophysiology, so in pathophysiology basically we had to study about the different factors that are affecting disease conditions, then about oxidative stress, then about various pathophysiology of depression, Alzheimer's, Parkinson's. So all that we study, right, in pharmacy about alpha-synuclein, beta amyloid plaques, tau proteins, neurofibrillary is just the basic. It's much more in detail. So how are these formed? How is there an inflammatory storm, neuroinflammation, excitotoxicity? There are various factors that affect these particular membrane permeability and all. So that is what was taught to us about these particular topics. Then about diabetes, diabetic neuropathy, complications of diabetes, nephropathy, retinopathy, rheumatoid arthritis, thyroid, so hypothyroidism, hyperthyroidism, drugs, rheumatoid arthritis, how is it being caused, TNF alpha, interleukins. So what is the role of different rheumatic bodies, rheumatoid nodules, rheumatic factors that are causing? Then, so this was the basics of our general of pathophysiology. If we talk about general pharmacology, so we were talk more. There are majorly many receptors, four types, but GPCR and tyrosine kinase. These are the two major receptors that are involved in almost every diseases, because PI3K, AKT, JAK-STAT pathway, tyrosine kinase pathway, protein kinase A, protein kinase C, cyclic AMP, DAG3AG, DAG-IP3. So these are certain common pathways that are involved in almost all many of the diseases. Then about transcription factors, so we have NF kappa beta, nuclear factor, NRF, that are involved in various transcription process. Then dose response relationship, so what is NOEL, LOEL, no adverse effect level, lowest observed adverse effect level, what is that sigmoidal curve, significance of the sigmoidal curve and all. Then we will talk about protein, G protein signaling, so how is G protein signaling actually being how what is the regulatory mechanism and all of the G protein signal, and a down regulation, up regulation, desensitization, resensitization of the GPCR receptors. Then some glimpse of nuclear receptors like estrogen, steroidal receptors and all was given. Next up, experimental pharmacology. So in this we were given information about the common laboratory animals that are currently available. Guinea pigs, rodents, non-rodents, different routes of administration, bleeding, how I mean that means blood sampling, anesthesia. Then we were spoken about the MCAO and the BCO, middle cerebral artery occlusion, and the brain carotid artery occlusion. So about cannulation techniques basically. Then the behavioral experimentation, like we have allodynia assays, we have behavioral assays, and then we have how to remove the biasness, how do you bring about randomization, replication, and all. Then drug solution preparation. So how do you prepare drug solution? What are the different factors that you have to consider while you're preparing drug solution? False positive, false negative results. Electrophoresis techniques. So protein and gel electrophoresis was taught to us. Flow cytometry, AI, role of AI, because this is a new topic that is included in the syllabus in drug discovery. Publication ethics and research ethics, what is what is the impact factor, what is the citation index, what is H index, I10. So what is plagiarism, how do you avoid, how do you select a journal, what are the different instruction to the authors, what is shimago ranking and all. There are so many different aspects while you're actually publishing your work, right? In vitro assays, cell viability assays, tissue fixations, imaging techniques like positron emission, positron emission, topography, and then tomography. Then there is SPECT, that is single positron emission computed tomography, cell counters, tunnel assays, and different fixation techniques and all. These were being taught to us. Next up, we have chemotherapy. So in chemotherapy, we were spoken about the different aspects of how is a disease occurring, what are the different chemotherapeutic agents. So bacterial, viral, fungal, and parasitic. These are the four major domains. If we talk about in parasite, amoebiasis, malaria, leishmaniasis. In fungal, we have candidiasis, aspergillosis. In viral, we have HIV, SARS like. In in bacteria, we have TB, pneumonia, and UTI, urinary tract infections. So a brief introduction about these diseases. How do community acquired diseases? What are the traveling diseases like traveler's diarrhea, motion sickness and all? So how do we treat them? TB may, what is the DOTS therapy? What is the treatment regime? Novel targets, and so that we get what is a drug pipeline and why are there not being drugs that are being more introduced. CP, so credit presentation is basically you get a particular topic on which you have to make a presentation and share it. So it imparts three skills within you: research skills, you learn how do you find papers, how many years back ka papers is you should find, what are the different journals from which you can find, how do you cite, you learn about citations, you learn about writing, how do we write the scientific language, when you present it shows your confidence, you learn about presenting your particular presentation in front of many people, so it helps in brainstorming as well. Labs, so labs basically were there to bring about a practical knowledge of what we are learning in theory. So electrophoresis, if we have learned in theory, practically how do we connect electrodes, how is there the separation of proteins based upon the molecular weights, and based upon the charges, what is stacking buffer, what is running buffer in biotechnology if I talk about. And then microtome, how do we process the tissue, what are the different staining techniques, how do we mount the tissue, how do we embed it, how do we perform GOD-POD, glucose oxidase peroxidase method, how do we perform Bradford, how do we perform HbA1c, and there are different techniques for protein glucose estimation both. So Fallencio Calteo, what is Lorry reagent and all. So these are all my credentials that you can connect with me, or you can connect with me via my Instagram, YouTube channel. I have a LinkedIn profile as well. You can join into our Telegram channel and you can follow me on these research sites as well. And I'll be sharing across a link as well in the description section whereby you can uh, track my recent updates. So thank you so much everybody for watching the video.